And our next speaker is Gabrielle Clark, who is a research assistant with the University of Dalhousie in the Mahar Lab. And Gabrielle will be talking about long-term enrichment effects on stereotypic behavior in farm mink.
Great. Thank you so much for attending. My name is Gabrielle Clark, and I'm going to be presenting a little bit of my master's thesis research that was recently concluded. And in this piece of research, we are looking at whether short-term juvenile enrichment could attenuate long-term stereotypic behavior in American mink that are farmed for their fur. So to give some background initially, stereotypic behavior, which in the welfare field is defined as invariant repetitive behaviors with no apparent function, are a prevalent welfare issue in fur-farmed American mink. In a large-scale Netherlands study, they were estimated to occur in 35 to 85% of adult female mink. And in a study observing a total or taking stereotypic behavior as a total observation period, they found that it occupied 32% of that observation period. So it can occupy quite a lot of their time that could be dedicated to other behaviors instead. As you can see, just in this row alone, there is a lot of individual variability in the types of behaviors that will be established. There are locomotor forms where all four of the feet are moving and the mink is moving along maybe the cage wall. There are also whole body stationary forms where the back feet might be planted and the upper body or front limbs are moving. They can also be isolated to only the head, or they can be oral forms like chewing on the bars or the wires, and they can also occur as scrabbling or digging against the cage wall. And some also consider fur chewing or tail chewing to be a stereotypic behavior, where others consider them to be abnormal repetitive behaviors. And in Canada and the US, one environmental enrichment is required in each pen to combat these behaviors. And usually it is either a standard plastic PVC ring or a golf ball, from what I've seen and what I've heard of in North America. And this enrichment came to be required in pens through research showing that additional enrichment or resources in pens can reduce stereotypic behavior or prevent its development. And that research shows that things like nest boxes, water for swimming, objects to chase and chew, or shelves and elevated platforms for mink to rest on can all reduce those behaviors. And that is above a barren environment or above standard enrichment. And at the neural or brain level, these enrichments are thought to have their effect through promoting the development or enhancing development of brain areas that control the goal-directed behaviors, rather than allowing the automatic habitual behaviors to occur more frequently, which is where the stereotypic behaviors would come in. And in mink, although there hasn't been a lot of research on the brain areas themselves, as a behavioral measure, rearing and enrichment is shown to promote behavioral diversity and flexibility in choice tasks. So there are a lot of problems with enrichment on fur farms, the standard enrichments, of course. So first of all, enrichment novelty is usually not renewed on farms. The guidelines say that when enrichment has to be provided, it's usually provided in early life, and then that enrichment remains in the pen for the rest of that mink's lifetime. So their use of it decreases drastically over time as they become habituated to it. And also the different subtypes of stereotypic behavior, like the types I talked about before, the locomotor forms versus the whole body forms and the scrabbling, they show different responsiveness to treatment with enrichment. So enrichment is not a one-size-fits-all treatment. And then also the tail chewing forms differ in their treatment from the locomotor and scrabbling forms. And then also the exact window when enrichment is most beneficial for mink has not yet been identified. So the guidelines say to provide it in early life. The farms can kind of interpret that differently between locations. And then if you wait too long and the stereotypic behaviors become established, they are then harder to treat with enrichment because they're more centrally controlled. So in our research, we aim to investigate whether housing juveniles with more numerous and more diverse environmental enrichments only during the juvenile period, and then removing those and leaving a standard enrichment, would attenuate long-term stereotypic behavior after 1 year in that standard enrichment. And we hypothesized that mink reared with these increased enrichments would show reduced adult development of stereotypic behavior compared to the mink reared only with standard enrichment. And I have an asterisk here because in another piece of our research that was published earlier on, we showed that this same group of juvenile mink actually had significantly more interaction with these enrichments. So if we assume that Increased interaction with enrichment yields more benefit. We hypothesized that these mink would show greater benefits of enrichment later in life. So for our housing conditions, we have the enriched kits group, which are the group that had greater enrichment in early life. This enrichment was in place from 4 to 15 weeks of age. So 4 weeks of age is also earlier than farms usually provide the enrichment. And in the picture on the right, you can see examples of some of the enrichment the mink might have had. So they had a hanging enrichment, which was either a sisal rope or a plastic chain, and that was exchanged monthly to maintain novelty. And then for the mobile enrichments, they had the standard plastic ring, which all mink had, and then an additional mobile enrichment that was exchanged every 2 weeks to maintain novelty. So it was either a hockey ball, a golf ball, a wiffle ball, or a pig's ear. And on the left, I have a figure of the cycle of production for fur farming. This one is for foxes, but it is very similar for mink. So they're born in the early spring. They're considered juvenile through spring and summer. And then in the border kind of between summer and fall is when they can be considered subadult. And this is when we removed those additional EK enrichments to really isolate that early juvenile period. And then we had a standard housed group, which only received the standard ring enrichment through that period and beyond, and a group that was enriched at whelping. So in this group, their mothers received enrichment, and that is not relevant for this study here. And we can consider them standard housed for the juvenile portion, since they also only had the standard ring enrichment from 4 to 15 weeks of age and onward. So for our behavioral sampling, we conducted scan sampling instantaneously of stereotypic behavior in the females that were remaining in the study at 16 months of age. And these samples were collected in October and November over 5 days. Our ethogram is on the right there, and we separated stereotypic behavior into locomotor forms, whole body stationary forms, and head-based forms. And we also looked for scrabbling and wire gnawing. Our sample size for each group was roughly 30 mink. We formatted these behavioral data as average proportions of observations where the behaviors were observed. And we analyzed them using Kruskal-Wallis one-way non-parametric ANOVA because the data were not normally distributed. And our significance level was 0.05. So contrary to what we hypothesized, we actually did not observe a significant difference between groups in development of stereotypic behavior. So on the y-axis here, there's the proportion of observations where the behavior occurred, which can also be interpreted as a percentage. And on the x-axis are the 3 different groups. So in the figure on the left, figure A, you can see the locomotor forms of the behavior. And then on the right, there is the whole body stationary forms. We actually didn't observe head-based forms, wire gnawing, or scrabbling in our group, so those aren't presented here. And then the plus signs actually indicate the means. So you can see that the mean performance of each behavior was relatively similar across groups. In the locomotor category, it was about 20% in each group of our observations. But interestingly, in the whole body category, the interquartile range was larger for that enriched kits group, which still does not give us a significant difference, but it does numerically indicate that the middle 50th percentile of those mink showed more variable performance of the whole body stereotypic behavior, which indicated to us that these mink might have developed more variable forms of stereotypic behavior than the mink in the other groups. So to jump into some discussion, first of all, there were some limitations to acknowledge in the study. Our sample size was reduced from our original group of 608 juvenile mink across the 3 groups to 104 adults remaining. So it would have been ideal to maybe follow more of those mink through to adulthood and increase our sample size to obtain more statistical power. And then also we only conducted the behavioral observations over 5 days. So it was 4 scans per day. So we got about 20 scans per mink, and that could have also been increased. So it's possible here that there might have been a false negative where if there was a difference in stereotypic behavior between groups, we may have been unable to detect it. But if we are to accept those results and try to interpret them a little bit or speculate about them, for the hypothesized effect in the EK group, it's possible we didn't find it because there could have been an effect of enrichment removal, which in species like primates, pigs, and mice has been shown to make stereotypic behavior worse. And that is thought to be due to a negative contrast effect, where if you take an animal that it is in an environment with a lot of behavioral opportunity and a lot of enrichment and place them in an environment with fewer relative opportunities, they may experience frustration. And then in the reverse, a positive contrast would be placing them in an environment that is comparatively better to their first environment. So frustration is thought to be one of the reasons that mink might perform stereotypic behavior. So it's possible that any frustration experienced by enrichment removal may have negated the beneficial effects of enrichment earlier in life. And then also, because we removed enrichments for this group around 4 months of age, it's possible that we missed a critical intervention age with enrichment. So in prior mink research, in one study, for example, where mink were reared with enrichment and then it was removed around 7 months of age, after 5 weeks, they did not show an increase in stereotypic behavior and they still remained at a lower level than the mink that were not reared in enrichment. And in another study, they provided enrichment to adult females on 2 farms that were around 7 months of age. And on one farm, their stereotypic behavior did reduce, where on another farm, they did not. So at this age alone, the enrichment has variable effectiveness. And that indicated to us that 7 months of age might be a critical age for intervention with enrichment. And that's also the age that stereotypic behavior is thought to become fully developed in mink. So overall, we did not find that additional enrichment as juveniles could attenuate stereotypic behavior after 1 year of enrichment removal and living only in standard enrichment. But in future research, it might be valuable to keep that enrichment in until about 6 or 7 months of age before removal to see if that is a critical window of intervention. And this is valuable for farmed carnivores as well as carnivores kept in captivity, like in zoo settings, because they also do perform stereotypic behaviors in these settings. And it would help us to better understand how to treat these behaviors with enrichment and how they develop. So this research has been published recently in the Animal Welfare Journal if you'd like to read any more about the methods and results. And thank you very much for watching.